week, but one exam will be on the courses, and the other exam will be a more general exam. So, I mean, I guess it's something to know about, and if there's some time conflict, make sure you're available during those times.
The general exam, is it Thursday?
Let me look on the schedule. No, so if I remember correctly, the general exam is first, so it's Thursday, and then the mini course exam, and the course on the mini course exam on the mini courses would be Friday.
How do you calculate our grade in that?
So, okay, so I'll say a little bit about the question. So I should have said this before. So each of the person who give a mini course will write the question. I guess Bill will not, but Ricardo will do the biological one. And those will be graded, okay? And the big exam, there are 4 people, 1 person from perimeter, I wrote one, Pedro wrote one, so there are 4 general questions, and we grade the questions. So you can look at all the exams from the previous years are all online, so you can see what kinds of questions are asked. I mean, obviously not the same questions, but at least the difficulty should be similar. Okay, any other questions?
Well, let me just add something, that the exam is extremely useful as a learning tool as well. The exam is very hard, it's long, just sitting for those 3 hours thinking about it and trying to solve it, you will learn more than in the lecture. It's really a very useful output of a learning tool. I think you will have fun doing it.
Okay, but eat well before, and so you don't get hungry during the exam because it's a long exam. Okay, so it's a pleasure to have Bill Bialek here. He gave some lectures last week already, and he agreed to give an extra lecture here today for you guys. So lucky you.
The very first course that I walked into as a student at Berkeley, which is where we first overlapped, the professor said, there are 3 ways to learn in the course. I'll be lecturing, we have an excellent textbook, and anything that's not covered in the lectures of the textbook will be on the final exam. So, so I don't think that's the principle here, but it is true that if we're going to make you sit there for 3 hours, you should learn something, right? I think that's an important idea. Yeah. Okay. So I want to talk about the physics of biological systems. For some of you, this will be an entirely new topic. I have very little idea. I'm not going to rely on specialized knowledge about biology. I think, today a high school course in biology has things in it that were university courses 20 years ago. So, and in any case, I think by the time you get to this level of your education, the idea that you should learn all the prerequisites before you start a subject is probably expired. I mean, that's not going to work. If you insist that you know everything you need to know in order to get started working on a problem, that's a good way never to get started, because you never know enough. So we're kind of just going to plunge in. But I do want to make one historical observation. I don't want to give a long discourse on the history of the subject, but it's important to know that the modern idea that there are separate subjects called physics and biology is a modern idea. It's largely a 20th century construct. So if you go back to the 19th century and you think about the great figures of classical physics, Helmholtz, Maxwell, Rayleigh, Ohm, all of these guys worked on problems that we would now separate and say, this is physics, this is chemistry, this is biology, and even this is psychology. So, for example, all of those people were very interested in how we perceive the world. And, for example, Rayleigh, right after he finished writing the great volume on the theory of sound, spent time trying to understand things about how we perceive sounds, and in particular, how we know which direction the sound is coming from. So just to give you a flavor, he understood, of course, because he developed the theory, that if you're listening to a sound which is at a high frequency, the wavelengths are short, and they're shorter than the size of your head. And so you can think in geometrical terms, right? Trace the rays. And that means that your head casts a shadow, or the acoustic equivalent of a shadow. So if a sound is coming from over here, it's actually louder in your right ear than in your left ear. But and so you can hear where it's coming from by measuring the difference in loudness or intensity of the sound. And you do. At low frequencies, this doesn't work, right? Because if the wavelength is longer than the size of your head, then there are no shadows. You're outside the geometric optics limit, right? And the only thing that you have to go on is that the sound will arrive at the closer ear before it arrives at the further ear. And so if you're listening to a single frequency, the only cue you have as to where it's coming from at low frequencies is the phase difference between the two ears. Now, at the time that he realized this, there was an overwhelming belief that we are phase deaf. And the easy way to convince yourself of that is to take a collection of tones and change their phases and ask whether you can hear the difference. And mostly you can't. That actually has a name, the fact that we are phase deaf. It's called Ohm's law. It's the same ohm. Different law, same ohm. And as with the other Ohm's law, it's only approximately true. And importantly, it's not true in comparing the sound between your two ears. If that wasn't worrisome enough, you can do a very simple calculation, which is that if you're looking straight ahead and you close your eyes and you listen for a sound and you ask, what's the smallest difference in the direction of the sound that I can distinguish? So I don't know where I can't see where it's coming from, but I can hear the difference. The answer is a difference of about a degree in angle. That corresponds to a difference in time between your two ears of only a few microseconds. And if you were a barn owl, you could do better. You could do 1 microsecond. The barn owls make their living by hearing the mouse rustling on the ground and not be at night, right? So they can't see them. So they have to hear where they're coming from and swoop down to get them. So Rayleigh actually went and did an experiment, which was to ask his wife, Lady Rayleigh, right? These were British aristocracy, to go into the gazebo with 2 tubes coming out of her ears, and which come back to a single source, and he makes a little sort of spark-like pulse, and then you change the length of the tubes and ask if she can hear the difference. Yes, you can. So this is all happening around 1900. Okay? So this is an example where physicists were interested in how our brains work, right, and how our ears work, how our senses work, because these are the physical instruments with which we use to observe the world. And so if, as a physicist, right, you're interested in how we know about the world, well, our eyes and our ears are how we know about the world, so you should be interested in how they work. This is a deeper example, I think, because it shows you that by taking some very simple facts about biology, the fact that you can actually hear where a sound is coming from at low frequencies, and then think about the physics that stands behind those facts, you realize that there's a problem. And that realization that there was a problem led immediately to an experimental question, and in fact, it launched a whole field which took 50 years to unfold until you could actually find the place in the brain where the timing comparison between your two ears is being made and show that it's made with microsecond precision. So the interaction between physics and biology is very old, but in the 20th century, the subjects diverged. And I think the view was that the prevailing view was that the relationship was one of application. So biologists had questions about how their part of the world worked, and physicists had tools, and you could apply these tools to help answer those questions. Now, I don't actually think this is fair to what was happening in much of the 20th century. It misses the fact that physicists, when faced with biological phenomena, would ask different questions than the biologists asked. And that actually drove a lot of developments in the 20th century. But sociologically, what happened was that that had its impact primarily in the biology community. So famously, the founders of what we now call molecular biology, many of them came from physics. And their impact was felt outside of physics. The physics community appreciated that, you know, we produce these smart people and they go off and they revolutionize other fields. It feeds our sense of our place in the hierarchy of disciplines, but didn't have much impact on physics. So something changed in, let's say, the last quarter or even the last decades of the 20th century, coming into our century, where the physics community started to embrace some part of all this activity at the border as being part of physics itself. And I'm not a historian, I'm not a sociologist, but you know, I've talked to lots of people. I mean, I lived through that transition, and I've talked to people who are, you know, my seniors, and seeing how things evolved. And I think one of the really important things that changed is that when I started out, if you asked physicists about biology, they would say, yeah, sure, it looks really interesting. I mean, you don't need an explanation for why the phenomena in biology are interesting, right? They're all around you. But it looks like a mess. And the closer you look, the messier it gets. So if you pick up a biology textbook and you're faced with just endless facts, and then you try going to a seminar to see what somebody did, and you look at the experiments they did, and you sort of think, no, particularly as a theorist, there's no way anything that I do as a theorist is ever going to connect with experiments that are that qualitative and messy. Okay? And unfortunately, the biologists sort of reinforced this in saying that this messiness was somehow intrinsic to biology itself. It wasn't just that they weren't trying hard enough to do quantitative experiments. It's that somehow there's a kind of irreducible sloppiness to the way in which life works. And what's changed dramatically in the last decades is that the part of the biological world, part of the living world, where our experimentalist friends can do experiments that look and feel like physics experiments. They're precise, they're reproducible, they uncover quantitative patterns in the data that as a theorist you look at and scratch your head and say, yeah, okay, this is something that needs explaining. That part of the living world has expanded enormously. So from the behaviors of single molecules all the way up to the behaviors of large groups of organisms, you have many examples where people are able to do experiments that have the look and feel of physics experiments. And so you feel like now you can plunge in and do something, okay? So what I want to do today, I don't want to spend a lot of time on telling you about all the different kinds of experiments people can do. First of all, I'm a theorist, so you'd get maybe, let's say, the artist's rendering of these things, not the real thing. Second of all, this is a theoretical physics school, so you didn't come here to learn about experiments. But it is important to understand, right? Theoretical physics works because at the end of the day, you compute numbers and people go out and measure numbers and you get them right or wrong, okay? And as sophisticated as we are as theorists, don't forget that that's why the enterprise exists, right? You really get the numbers right. The astonishing thing is not that we study things of great mathematical beauty. It's that those structures of great mathematical beauty describe the real world. That's the surprise. But you only know that if you can do experiments that are at a certain level of precision. So what I want to do today is to talk about two broad themes in the physics of biological systems, which illustrate these ideas. And I'm going to give you sort of a little bit of several things, in part because part of the point of the school is you might want to come spend time with us, and you should know that even one of us, with help, works on a wide range of different problems. So the physics of biological systems is not one thing, it's many things. And what this means is that my agenda for this period is rather flexible. So I'm going to talk about several different things. If something's confusing, please interrupt, because I don't know what you guys know, and I might use language that is a little jargony. I try to avoid it. But stop me if I don't. As one of my colleagues likes to put it, if you're confused about something, there's probably somebody else in the room who's also confused about it. So one of you should be brave and interrupt. So the two main themes are precision and emergence. So emergence is the more obvious one. This is the statement that life is more than the sum of its parts. So throughout the 20th century, one of the great triumphs was if you want, the export of the physicist reductionist view of the world. Let's try to find the fundamental constituents of matter, to the phenomena of life. Let's try to find the fundamental constituents of life. What are the molecules that actually make life work? And we have now an essentially complete catalog of those molecules. An astonishing thing to be able to say. And despite the fact that one often thinks of biology as focusing on diversity, right? Every organism is different, that's what's interesting, that's what's important. To an amazing extent, the molecules of life are universal. So a dramatic example of this is that if you want to do something with your DNA, it's useful to take a little bit of DNA and make more of it. So you should make lots of copies of the DNA. So how do you do this? You use a molecule called DNA polymerase, which is the protein inside all cells, which has the job of copying your DNA. That's what happens every time a cell divides and so on. But actually, there's a trick, which is if you want to make lots of it, you should do it in a chain reaction, where one molecule gets copied and it becomes two, and then the two get copied again, and so on. And in order to make this chain reaction work, it's useful to be able to change the temperature of the system by a lot. So if you heat up DNA above the temperature where it normally lives, then the two strands of DNA will separate. Now you can copy each one. So normally inside the cell, right, the DNA is wrapped up, and copying it is an incredibly complicated topological problem, right? Which, by the way, Watson and Crick identified when they proposed the double helix structure. They noticed that there was a problem, and that became, again, a whole field. There's a whole collection of molecules that are responsible for regulating the topology of DNA that get engaged when you want to do this copying. But if you want to do it in the lab, it would be easier if you could short circuit this. So what you do is you just heat the thing up and the DNA comes apart. And then you let the polymerase work, and now you cool it back down and the molecules come together, and you do it again, and so on. The only problem is that most proteins, if you put them in the solution that you've heated up to the point where the DNA melts, the protein also melts and it's dead. This is how you cook, right? Meat is mostly protein. If you do this, you cook the meat, right? So you all know this. So what do? You go find an organism that lives at very high temperatures. So you go to the bottom of the trenches, the deepest trenches in the ocean, where the thermal vents are. And you find an organism that lives, well, also at very high pressure, because it's at the bottom of the ocean, but at very high temperature, because it's right next to one of these thermal vents. And of course, the DNA polymerase inside that organism doesn't mind being at a very high temperature. It doesn't get cooked. Now, so one physics problem is, how do you make a molecule that doesn't get cooked when you heat it up that much? Okay. Wonderful problem, I'm not going to talk about that. The reason I brought this up is that you can reach into that bacterium that lives at the bottom of the ocean, which has never had any contact with humans over the billion years of evolution of both of us. And you can take the DNA polymerase out of that and copy human DNA, and it works. So part of the reductionist program was the discovery of these molecular components which are universal enough to be interchangeable. It's not like electrons, right? Which really are all alike. But these are close enough that they're interchangeable. So we know all the parts, but we also know that a single molecule is not alive. And any of the things that you see in the living world that attract your attention almost surely are the result of interactions among large numbers of components. So in order for even to make a single molecule that does something as complicated as copying DNA, that molecule is a polymer that has hundreds, if not thousands, of individual elements. And so the structure and function of that single molecule emerges from the interactions among hundreds of the amino acids that are its constituent parts. And the functional behaviors of cells emerge from interactions of hundreds and thousands of different kinds of molecules. The things that you're able to do right now when you're listening to me emerge from the coordinated activity of thousands or even millions of neurons in your brain. And many of the things that you see every day around you are the result of interactions of hundreds, thousands, millions of organisms. And there are beautiful isolated examples of this, like a flock of birds or a swarm of insects or a school of fish. But of course, there are also more real social examples, like in our construction of cities or in termites' construction of a nest or something like that. So on every scale, from the molecular to the ecological, there are emergent phenomena that come from the interactions among large numbers of constituents. And of course, we're used to emergent phenomena in physics. This is what statistical physics and condensed matter physics are all about. And there's been a long dream that the language of statistical physics would also be a language for describing emergent phenomena in biology. And so I'm going to tell you a little bit about progress in this direction. And you can, as I say, you can think about this at the level of molecules, at the level of cells, the level of neurons, at the level of populations of organisms. And okay, and again, this subject is much more exciting today than it was 10 years ago because of dramatic improvements in our ability to do experiments on all these different scales. Okay. Now, there's a more subtle thing, which is not so obvious. And that is that contrary to the impression that biology is messy and sloppy, and of course aspects of our lives are messy and sloppy, and some of us even enjoy that, is that biological systems are capable of extraordinarily precise behavior. And the classic example of this is the ability of your visual system to count single photons on a dark night. So if you're outside and it's dark, let's say dim moonlight, not too many stars, you're capable of raising your hand and saying you saw something when 5 photons arrive at your retina. And in fact, the reason it's 5 is because there's a little bit of dark noise in the background, and so you just want to be sure. If we do the experiment right, can force you to report the individual photon responses. And again, this is a long saga. It begins in 1911 when Lorentz, he was finished with transformations, realized that the measurements on the dimmest light that people could see was consistent with the idea that there were not very many photons. I mean, the numbers weren't so accurate. It was hard to estimate. So he asked his colleagues, is it possible that the answer is that you can count single photons? And that, again, started a whole series of things which culminated with experiments on human observers showing that that was true. That happened in the 1940s. And it took until the late 1970s until you could actually measure directly the responses of individual cells to single photons. Okay? But they're there, and we now understand something about the molecules that are involved in making that happen and so on. But photon counting isn't the only example. I told you about microsecond precision in measuring the timing difference between your 2 ears. You probably know that bats navigate the world by doing echolocation. They make pulses and listen for the echoes. They can measure the timing of the echoes to an accuracy of 10 nanoseconds, which is, I think, the most extraordinary number in biology. When you think that all of the things that happen in the nervous system happen on a timescale of milliseconds, microseconds is already hard to understand, and 10 nanoseconds is, well, 100 times harder to understand. And actually, if you think about it, if you're asking about your ability to measure something against the background of noise, right? It's really it's the square that matters, right? Because you're averaging over random things. So you could argue that 10 nanoseconds is not 100 times harder than 1 microsecond. It's 10,000 times harder. In any case, it's an extraordinary number. There are many examples where biological systems use changes in the concentration of molecules as the basic signaling mechanism. So the signal that tells a cell what part of the body it's going to become during embryonic development is the concentrations of some particular molecules, and I'll talk more about that in a moment. And there are several examples where you can convince yourself that the precision with which the system is responding to those changes in concentration is essentially so high that the system must be counting every single molecule that arrives at its target. Because after all, molecules arrive at random. Right? There are higher order examples. There are not just, in the nervous system, you think not just about seeing that something is there or pointing to where the sound is coming from, but more complicated things like extracting features from what you're looking at. How fast is that object moving across my visual field because I want to reach out and grab it? And there are examples, for instance, where the precision with which your brain can estimate, or the brains of different animals can estimate the velocity of movement of an object is so high that it approaches the limit that's set by photon shot noise in making the image. Okay. If you think about the problem of learning, you could say that learning, if you're in a probabilistic setting where the relationships among the things that you're trying to learn are not deterministic, then learning is a problem of inference of the underlying probabilistic rules. And your ability to infer probabilistic rules is limited by the number of samples that you've collected. And there are examples in which the learning by humans and animals approaches that limit. That is to say, the reliability with which you've inferred the underlying probabilistic rule is as high as it can be, given that you've only collected a limited number of samples. Okay. There are more subtle problems like, in order to actually transmit information from one place to another, I need to go through some limited physical device. So for instance, if I'm a cell and I want to change the concentration of one molecule and have that have an impact on how many molecules of some protein I synthesize, well, what happens? I change the concentration of this molecule binds to the DNA. By binding to the DNA, it triggers some process by which the messenger RNA gets transcribed, that gets translated to the ribosomes, and I make the protein. I'd like to be able to take this concentration up and down and have this other concentration follow it. Well, you can think of that as being a communication channel, right? I put a signal in at this end, and out comes something at this end. Well, like all communication channels, it's a physical device. It's a physical device made out of a surprisingly limited number of molecules. Individual molecules behave at random, so there's some noise level that you can't get below because of the finite number of molecules. That means that this communication channel has a limited capacity to transmit information. And there are examples in which the real system is approaching that limiting capacity, which is set by very basic physical principles. So what my colleagues and I have been interested in on this side is both trying to collect these examples and trying to turn this around and ask, is the fact that real biological systems approach these physical limits a path into developing theory of how these systems must be built. So I'm going to start by talking about some examples here, and then I'll shift over to here. And the reason I want to start here is that this is also an opportunity to emphasize what's happened with the quality of experiments. Because you can't discover that systems are precise by accident, right? You have to make a precise measurement. So you can't have this discussion unless the experiments are good enough. So let me start with an example. Suppose that you look at a caterpillar, so a baby insect. If you look at a caterpillar, I'm a terrible artist, so let me. Okay, so there's a caterpillar, and you've all seen these. And you notice that there's a very obvious feature of the caterpillar, which is that the body comes in segments. And in fact, to a remarkable extent, these segments are very similar to each other. Now, our body comes in segments too. Think about your vertebra along your backbone. But it's a little less obvious in us than it is in insects. You can see it on the outside, okay? So you might ask yourself, how does the animal know? Remember that right, all animals start, us included, as a single cell, fertilized egg. The cell duplicates. You eventually have 2 to the n cells, where n is some reasonable number. And then the cells start to become different parts of the body. They move around. And lo and behold, at the end, you have segments. So where in that process of cells dividing and deciding to do different things, where do these lines get drawn? And how does the animal know where to put them? So the answer is a little bit different if you look across all animals, there's a lot of variety. If you look across insects, there's less variety. There are some things that are universal. There's one insect in particular that is very much beloved of the biologists because it's a fantastic model system, and that's the fruit fly, which you can presumably find in countries that have great fruit. Just leave it outside to rot for a little while, and you'll have a collection of fruit flies. And the reason fruit flies are a favorite of the biologists is they reproduce quickly, so you can do genetics, you can breed them. And the astonishing thing is that when the mother fruit fly lays an egg, the little caterpillar, which for the case of flies is called a maggot, less attractive. So think about caterpillars. It's a fruit fly, right? So it doesn't have anything to do with decaying flesh, okay? Which is our usual association of maggots, which is why we don't like them. But anyhow, so think about caterpillars, but in practice it'll be a fruit fly larva. Mother lays an egg, and the caterpillar walks away 1 day later. So the whole process from one egg to a fully formed organism that's walking away, and by the way, a larval insect, as soon as it walks away, it starts eating, right? That's its whole job in life. So fully functional, right? 1 day. In fact, it's even more impressive than that. The steps where you go from 1 cell, 2 cells, 4 cells, 8 cells, they happen extraordinarily fast in the fruit fly at the beginning. In fact, they happen so fast that what actually happens is that the nucleus of the cell, which contains the DNA, duplicates, and you copy the DNA. But you don't stop to build membranes that separate the cells. And after about 9 or 10 rounds of this, almost all of the cells move to the surface of the egg. So what you have is an egg which is about 0.5 millimeter long. And the cells are all on the surface. So if I make a slice through the egg, the nuclei are all around, right? And if you know which molecule to look for, you can find that there's a particular molecule whose concentration you can measure, and we can talk a little bit about how you do this if you're interested. And if you measure the concentration, it's a molecule that goes into the nuclei, so you should think of it as concentration in the nuclei, as we close. And if you measure that concentration, it looks like this. And in fact, there's a few of these molecules. So there's another one. It looks like that. And I think you can imagine what the other ones look like. And if you count the stripes and you count the segments of the body, you start to see how this works. And if you come away thinking that the answer is, well, the stripes are the blueprint for making the segments, that's not exactly right. It's a little bit more complicated than that, but it's close. And in particular, if you watch the embryo, oh, I forgot to tell you something. You can measure this after 3 hours. So in 3 hours, the embryo has laid down the whole blueprint for how the body is going to be built. And every cell at that point knows what it's supposed to do. Okay, so I know what these molecules are. I even know how they get here, because that's in the biology textbooks. So what's the problem? In particular, what's the physics problem? So in order to understand what the physics problem is, you have to make a quantitative measurement. So let's ask, let me look at 100 embryos. And let me measure the positions of these peaks in 100 embryos. How much do they vary? The answer is that the variation in the position from embryo to embryo is less than 1% of the length of the embryo. And this is if you use flies that are very closely related to each other, so there's not much genetic diversity. If you grab the flies from outside, of course they'll be different, but it's also true that we all look different. So that doesn't represent imprecision, that represents genetic variation. Okay? So you want to ask this question in something that's as close as possible to genetically identical flies. So the program that leads to building this blueprint is precise at the 1% level. How does this arise? Okay, actually, let me tell you something else. You might think, okay, where does this pattern come from? So you say, it's a periodic pattern, almost. So maybe this is like other problems that I know about in non-equilibrium physics, right? I have a box. I have some dynamical process going on that's not at equilibrium. The uniform state becomes unstable, and I get a periodic pattern emerging. So this actually was Alan Turing's idea for how embryonic development worked, and he wrote this down in 1952, shortly before he died. So this is the same Turing of the Turing machine, right? The same Turing of the Turing test in artificial intelligence. And, well, it is sadly worth remembering that he lived only half a life, right? So imagine what we lost, but anyhow. Unfortunately, what Turing couldn't possibly know was that that's not how it works. And that's not how it works, not because biology is more complicated than physicists and mathematicians would like. It's because the problem is different. It turns out that when the mother makes the egg, she breaks the symmetry at the beginning. So it's not the case that the embryo is this symmetric object in which all points are equivalent. From the very beginning, they're different. And they're different because what the mother does is to put, for example, I mean, there's a few of these signals, but let me just discuss one of them. Okay. Okay, I'm running out of colors. What the mother does is she puts the, it's okay. Oh, they're here, okay. Oh, right. The mother puts the messenger RNA molecule for one particular protein stuck at the end of the egg, which will become the head. So what happens? Well, once the egg is laid, the message begins to be translated. And so you have a source of proteins at this end. Those molecules can diffuse through the embryo, and they also, right, because there's no membranes dividing the cells yet. And so the protein can diffuse through the embryo, and it also gets degraded. So what does that mean? It means that if I measure the concentration of the protein, and I think about how it changes in time, it changes due to diffusion, but then it also decays with some typical lifetime tau. And there's a source at this end which means there's a boundary condition that there's flux coming into the box. So if you wait for a while, this will come to steady state. And you'll have an exponentially decaying profile. So go find out from your biologist friends what this molecule is, and go measure its concentration. Now at this point, I should tell you, okay, how the hell do you measure the concentration of this molecule inside the embryo? So there's two ways to do it. One is you purify this molecule, you inject it into a rabbit, and you make antibodies against this molecule. And then you tag the antibodies with a fluorescent tag. You take the egg and you stop everything by gently cooking the egg. And then you make holes in it and diffuse the antibodies in. And they'll stick to the protein, but they're carrying a fluorescent tag. So if you shine light and measure the intensity of the fluorescence that comes back, that tells you about the local concentration. Okay, that's actually a surprisingly precise method. Sounds bad because, I don't know, you cook the egg, you make holes in it, there's the rabbit with the antibodies. It sounds complicated, but actually, it's an incredibly precise measurement if you're careful. The other, more modern way to do it is to genetically engineer the fly. So you take the piece of DNA that codes for the protein that you're interested in, and then from a jellyfish, you get the piece of DNA that codes for the famous green fluorescent protein. And you put these next to each other, and you put that back into the organism. And now whenever the fly wants to make this protein, it keeps reading and makes the fluorescent protein, and they're stuck to each other. Now you have to do a whole bunch of experiments to show that when these things are stuck to each other, it still works the same way. Because after all, you're carrying around this other big protein. Okay, this is a theoretical physics school. I don't have to tell you how they do that. Okay, that one you can trust me on. So if you can do that, then you can measure this live. You can take the embryo, put it under a microscope, shine light on it, and measure the fluorescence at every single point. Okay. And indeed, you see an approximately exponential concentration profile. It turns out there are a couple of other molecules where the same idea works. There's another one that comes from the back, and there's a special one that's symmetric at the front and the back, and it defines the ends. Okay? But let's think about this one. So first of all, this is what tells you that the problem isn't the problem that Turing was worried about, right? It's not symmetric from the beginning. In fact, if you try to manipulate the system and change the concentration of this protein, then you move these segment boundaries around. So we know that the information is flowing from these inputs that the mother gives through, there's a big box in here. These molecules influence a whole set of other molecules, which eventually influence these. Okay? And it comes out in these beautiful stripes. So let me tell you another couple of quantitative facts. Okay, why did I bring up Turing? Well, first of all, it's interesting. It's an interesting example where you might say, well, I think I know what physics problem this is. It's like this other physics problem. And then you realize, no. It's not like that problem. There's a different problem here. And the way to convince you that it's a different problem is, suppose that you have a conventional non-equilibrium pattern forming system. For instance, suppose you have Rayleigh-Bernard convection. Right? So I take a thin layer of fluid, I put it between two plates, I get the bottom plate hot, the top plate cold. If the temperature difference is very small, then the heat is convected through the fluid doesn't move. If I increase the temperature difference, then there's an effect where the fluid at the bottom gets hot. When it gets hot, its density goes down, so it rises. But, well, what goes up must come down, so it must flow up and then come back. And what actually happens is you get these rolls of convection, right, where locally you have fluid going up and down, and there's a characteristic scale for these, and that pattern gets repeated. And so you can get a spatially periodic pattern just in your kitchen, right? I mean, you can do this experiment on your stovetop. It's not as controlled as when they do it in the lab, but you get the idea, right? And if you've ever flown in a small plane over the desert, you've also felt this, right? Because the atmosphere is being heated, well, you might think it's being heated from above by the sun, but of course it's the ground that absorbs the sunlight, so the atmosphere is being heated from below. As those of us who live in cities know very well, right? And so you get these convection rolls, and if you fly over them, the plane will actually bump, right, as you feel the rolls, which is sort of extraordinary. Anyhow, so that's the kind of thing that you might have thought was what's going on, but it's not because, well, it's not because Turing proposed that as a mechanism because he thought the problem was to spontaneously break the symmetry, but in fact the symmetry is broken at the beginning. But more importantly, but also more subtly, if you do Rayleigh-Bernard convection, what happens if you make a bigger box? Well, the answer is that the size of the convection rolls is set by parameters of the fluid. So if you make a bigger box, you get more rolls. What happens if I make a bigger box here? Well, that corresponds to the mother laying an egg which is bigger, which happens. The eggs are not all exactly the same size. If I make it 10% bigger, it's not that big a deal. If I take two eggs that differ by 10%, count the stripes, there's room to put in one more stripe. Never happens. The system makes 7 stripes. So now you ask, well, what's the physics problem here? Well, let me look at lots and lots of embryos all laid by the same mother, all of slightly different sizes. In fact, 10% is if your conditions are reasonably stable, 10% is a rather large variation. The eggs are reproducible in size, but they vary enough that you could fit in extra stripes or miss a stripe if it was too small. And what you can do is to show that if you look across large numbers of embryos and you measure this position and this position and this position, so x1, x2, x3, what you find is that the positions of these stripes are exactly proportional. The distance from the head is exactly proportional to the entire length. You have perfect scaling of the patterns. And again, that scaling is down to a precision of 1%. So this is an example of a pattern-forming system in which the pattern that's formed automatically rescales to the size of the box. That's a fantastic physics problem that I think nobody knows the answer to. Write down the simplest example. It would be nice to understand how this particular thing works, but write me an example of a pattern-forming system that automatically rescales to the size of the box. None of the non-equilibrium pattern-forming systems that we're used to do that. So there's a physics problem for you. And you would, because the variations in egg length in a genetically pure strain of flies, an inbred strain of flies, is so small, you would not be able to see this if the whole system weren't so precise. Yes?
Same question. If you put a theory without scale in a box, isn't that what typically will happen? Because there's no other scale?
Yes. So the problem is that sorry, so I guess I overstated the difficulty of the problem. It's very difficult to imagine a theory of molecules interacting with each other in which there's no scale. Because the only reason things move in space is because of diffusion. And there is some time scale on which chemical reactions occur. And so when you combine diffusion with those time scales, you get length scales. And it's hard to figure out how to get rid of them. But I think that's the right answer, is that somehow the network of reactions has to be tuned someplace where all those scales go away. But we don't have a concrete example of that. The other problem that arises here is that if you think this is the signal that ultimately drives things, then what that means is that two cells, which are right next to each other, which differ in position by roughly 1% of the length of the embryo, must be able to do different things, right? Because apparently there's enough precision to specify things to 1%. The distance to the next cell is actually a little more than 1% because there are fewer than 100 cells along the length. So that means every cell knows where it is. So if this is the signal, how big is the signal that is driving that variation? And the answer is it's a 10% difference in concentration. So that means that the system is capable of responding to 10% differences in concentration. So we don't know yet whether that's a lot or a little, right? Might be that that's easy, might be that it's hard. You might suspect from where this is going that it's going to turn out to be hard. But there's another part, which is that if you think that this is the origin of the positional information, and the system's capable of doing different things in response to 10% differences, then in order to make sure that two different embryos do the same thing, you better make sure that the concentration is the same to within 10%. And this was the place where all the biologists balked. So all of this stuff was in textbooks, and we pointed this out many years ago now, and our biologist friend said, no, that's not how it works. I don't care what it says in the textbooks, we must be missing something, because there's no way that the absolute concentration of a molecule is reproducible to 10% accuracy between two different organisms. But I told you that now you can measure, right? You make the fluorescent version of this, you look, you put two embryos side by side, and you take your laser scanner, right? And you go over here, and then you go over here, and you compare corresponding points, and you ask, is the concentration the same? Which is the question of whether the brightness of the fluorescence is the same. And the answer is it is. And you know how accurately it's the same? 10%. And actually, now you can do a better experiment, which is that you can grind up the embryo, extract these messenger RNA molecules, and count them, and ask how many the mother put there. And you can show that that number is reproducible from embryo to embryo to a little bit better than 10%. Just a picture of the mother making the egg, counting out these messenger RNA molecules to put them in. Why is 10% surprising? What's the smallest difference in concentration that you can actually measure? So this is a problem that's very similar to problems that we know about. If I ask you to measure the intensity of a light, I ask you how bright a light is, and I say, what's the smallest change in the brightness of the light, the intensity of the light that you can measure? You know the answer. You're counting photons. If it's an ordinary light source, then the statistics of photon counting is Poisson. So there's a square root of n, right? And that's the answer. What about molecules? Well, you don't really measure concentration. What you do is you have some detector which is of some size, and you count the molecules that arrive at that detector. So let's imagine, in this case, for instance, all of these proteins are proteins that affect the DNA. So the detector is some little piece of DNA where these molecules bind. So let's say it has some linear dimension A. Well, what are you doing? You're basically constructing a volume, and you have these molecules that are all over the place, and they're at concentration C in the bulk. And what you're doing is counting the number of molecules in here, and if you count them, then the average number you'll get will be the concentration times the volume in which you counted, because that's what concentration means, right? It's no magic here. The square root of n argument, right? The molecules, the reason that counting photons gives you a square root of n, okay, maybe you all know the reasons for that. There's two possible reasons. One is that you're looking at a laser and the light's in a coherent state, so intrinsically the number of photons is Poisson distributed. The other possibility is you have a very bright light source and you're only seeing a very small fraction of the photons. And so which fraction you see is chosen at random. And so that turns whatever process it was into a Poisson process. For molecules, it's also a Poisson process. And the reason is the individual molecules are moving around at random. In particular, if they're at low concentration, then they don't know about each other, so their positions can't be correlated. So they must be Poisson. So that means that you get fluctuations in the counting, which are of order square root of n. But since counting molecules is basically the same as measuring concentration, that means that the accuracy with which you're measuring concentration, the fractional accuracy, is 1 over that square root of n. But that's if you count once. What if you're willing to take time and count many times? Well, if you do it again immediately and count the same molecules over again, assuming that you didn't make any mistakes the first time, it doesn't help, right? It's the same molecules, it doesn't tell you anything new. What you have to do is to wait for the molecules that are in the neighborhood where you're looking to exchange with the rest of the solution. And after they exchange, you get a new sample. How long does that take? Well, it takes the time required for the molecules to diffuse in and out of this region. So there's a correlation time, which if you multiply it by the diffusion constant, you get the square of the linear dimension of your detector. Now, if I'm willing to wait for a total amount of time tau, then every tau c, I get a new sample. So the total number of samples I get is tau divided by tau c. And you know that if you get n independent samples, you should reduce your noise level by 1 over square root of n. So I should get this. And if you plug in all the factors, you get this combination, which you can easily see is dimensionally correct. And in fact, it's more or less the only dimensional. It's the obvious thing you would write down by dimensional analysis. It has a very simple interpretation. So it's the diffusion constant times the linear size times the concentration times tau. Well, imagine that every molecule that entered this region disappeared because you counted it. Okay. In truth, when the molecule enters the region, it has to do something in order to be counted. But let's not worry about that. Let's just imagine once it arrives, I take it out. Well, what's happening then? I have a big solution. I have a region of linear size A. And anytime a molecule enters that region, it disappears. So that means that the concentration at the surface of that region will go to 0. So I get the diffusion equation with a boundary condition at the surface of this region, whose linear size is A, where the concentration is 0. And then outside, far away, the concentration is C. So what's the concentration profile going to look like? Well, it's going to have a gradient at the surface, which is of order C over A. But if there's a gradient, then there's a flux. And indeed, right? Every time a molecule arrives, you take it out. So that's only possible if there's a continuous flux in. What's the size of the flux? It's the diffusion constant multiplied by the gradient. And then you integrate over the area. The gradient has a 1 over A, right? D times the gradient is C over A. So if I have linear dimensions A, if I think about the molecules all coming in, then at this point, the gradient of C will look like C over A. So the flux will be D times the gradient of C, which is dC over A. And then if you integrate over the surface, you get an A squared. So you get dCA. This combination is just the rate at which molecules are entering your detector. That's a rate you multiply by the time, you get the total number that you counted. And then the answer is your fractional error is 1 over the square root of the number of molecules that you counted. It's just like counting photons. So, 2 questions. One is, if you plug in the numbers, what happens? So I'll do that in just a second. The other question is, should you believe this back of the envelope argument? That's a much more interesting question in many ways, especially if you're theoretically inclined. So what many of us have done over the years is to try to do more and more realistic calculations, not of this stochastic process, but to remember, for instance, that if what's happening is that the molecule is binding to a binding site, then what you're really doing is monitoring the occupancy of the binding site, and that fluctuates. And you have to turn those fluctuations into an equivalent noise level for estimating the concentration. But that's an easy calculation because, or rather, there's a strategy for doing that calculation because the fluctuations in the occupancy of a binding site, if the system comes to equilibrium, those are just thermal fluctuations. So I can compute the power spectrum of the fluctuations in the occupancy of the binding site by using the fluctuation dissipation theorem. The coordinate is the occupancy of the binding site. The conjugate force is the energy difference between bound and unbound. I compute the response of the coordinate to the conjugate force. That gives me a response function. I plug it in the fluctuation dissipation theorem. I can compute the spectrum. I can then remember that when the molecule leaves, it's diffusing, so I should couple the binding and unbinding to the diffusion equation. Go through the calculation again. What's amazing is if you do that, you discover the noise has 2 terms, one of which is associated with the details of the rate constants for coming on and off, and the other of which is only coming from the diffusion constant, and it's this. And you can make the events that happen at the binding site more and more complicated, but you keep getting the term from diffusion, which is just this limit. Sometimes the role of theory is to show you that your rough estimate is correct. Okay. So numbers, if what you're doing is regulating what's called gene expression, so that is to say, these molecules bind to the DNA and they control whether the information that's coded in the DNA eventually gets read out to make proteins or not. That process is called gene expression. So we know something about the overall numbers. We know that we're talking about a protein. So that means that the diffusion constant inside the cell is micron squared per second. You can measure that. We know that it's binding to some small binding site. So in order to be specific, that binding site has to be the size of the molecule. So it's about 3 nanometers. We also know that the concentrations of the proteins that control whether you read out the information from your genes or not is surprisingly small. So the concentrations are measured in units of nanomolar, which is a unit that physicists don't have much affection for. This is inherited from the chemists. So let me remind you what this means. Nano is 10 to the minus 9, of course. Molar is Avogadro's number in a liter. But liters are also not a very useful unit for thinking about cells. Cells have dimensions of microns, so maybe it's useful to know that this turns into roughly 1 molecule per cubic micron. If you put these numbers together, you can see that DAC is about 1/300th of per second, which is about once every 5 minutes. So that's a sobering thought. These molecules are arriving at their target about once every 5 minutes. But I told you that the whole pattern is laid down within 3 hours. And in fact, the crucial part of this is all happening in about 10 minutes. So if you only have 10 minutes, then DAC tau is 2. And 1 over the square root of 2 is, 1 over the square root of 2. It's not 10%. It's basically order 1. So if you just take all the numbers at face value, you would conclude that it was impossible for cells to distinguish 10% differences in concentration. So of course, no laws of physics are being violated, right? But this tells you that you're missing something. And more importantly, as you try to think about, how as a physicist should I think about biological systems, what it tells you is that very fundamental sources of noise, in this case due to the random motions of individual molecules, are on a scale where they're comparable to the signals that biology is actually using in order to make decisions. Okay. So I'm not going to talk about exactly how does this all get resolved and everything. It's more to give you a sense of what the problems are. And in fact, this example tells you that the system is precise in the sort of kind of simple phenomenological sense. The idea that you can position things to 1% accuracy sounds impressive. If you think a little harder, you realize, well, there's fewer than 100 cells along the length, so that means that every cell kind of knows where it is. You can't do better than that, right? There's no space in between the cells, right? So you basically have a precision of estimating position, which is down to the level of the lattice spacing. This ultimately is driven, this also allows for scaling with very high precision. It's ultimately driven by inputs whose concentration must be measured to an accuracy which is such that it's close to the limits that come from counting molecules. In order for that to be meaningful, concentrations have to be reproducible on the same scale, and they are. And this makes you think that this whole process of going from these inputs all the way to these patterns has been selected for through evolution to get as much information from here to there as you can while using this very limited number of molecules. And so the theoretical problem that many of us are interested in is what constraints does that place on all the things that happen along this path? It's a good thing that the noise levels are low, but that's not the whole problem. So for example, at this stage, to get from here to there, the information is contained in the concentration of several different molecules that are sitting in some network in here that I haven't drawn. And you have to, the embryo has to kind of read that information out. So you could ask, if I measure the concentrations of these molecules and I measure their noise levels and all that, what is the best strategy for reading out position? And the answer is that if you use the best possible strategy for reading out position, right? This is a thing our experimentalist friends do. You're measuring something, the measurement is noisy, you characterize the noise, you have multiple measurements, you characterize not only their individual noises, but the correlations between them. You ask what's the best way of combining them in order to pull out the thing that you really care about. Do that calculation, but for the real data that comes out of the embryo. And the answer is that you can infer your position to 1% accuracy, which is the accuracy with which these stripes get made. So that tells you that there's just enough information at this level to specify them. But only if whatever happens between here and here effectively implements the optimal strategy for reading the information out. Along the way, we have to construct that optimal strategy. How do we know that we're right? Well, these are flies. You can make mutations that will manipulate these signals. Those propagate into here. They produce different combinations of the concentrations of these molecules. You feed those into your calculation for what the best position is, with best readout of position. And you end up predicting, for instance, that there's a cell that's sitting here that thinks it's there, and so it should make a stripe. And then you go and look, and it makes a stripe. No free parameters. The only idea is that what the embryo is doing is implementing the strategy for reading out the information that captures all of the information that's available at this level. So it's pushing to extract the thing that it cares about down to the limit that's set by the physical properties of the signals that it's using. Okay. So there are more and more examples of this flavor. Let me say one more thing, which is something that we're working on now, and then I'll switch from thinking about precision to thinking about emergence. When I told you that you know your position to 1% accuracy, and there are fewer than 100 rows of cells, so therefore every cell knows what it's doing, I lied a little bit. Let me expand everything for you. So there is the positions of the cells. And I told you that, and let's say this spacing, just to be concrete, this spacing is 1. In fact, we know it's roughly 1% of the length of the embryo. And I told you that the cells, by using these molecular signals, know their position to 1% accuracy. So that means that if you, as it were, interrogate this cell and ask it where it thinks it is, that is to say, you look at the concentrations of these molecules and you infer where it is as accurately as you can, your estimate will have this error bar, and this error bar, right, this is the distance between cells. There's something like 60 cells along, 60 rows of cells along the length. So this error bar is about a half in units where this is 1. So you say, that's great. My error bar is smaller than the distance to my neighbor, so I know where I am. But you realize, of course, that this isn't true. There's some probability that you get confused. There's a way of thinking about this which is very useful, which is suppose that I give you a number between 0 and L, and I choose the distribution to be uniform. But what I tell you is y, which is x plus some noise, where this is sigma squared. So you might think to yourself, well, I can distinguish L over sigma different possibilities, right? My basic scale is sigma. There's L things. So I can sort of make bins of size sigma. I have L over sigma of those. So if I ask you in bits, how much information does y provide about x? You're tempted to say the answer is that, right? It's not quite true. What's the information? The information is the difference between two entropies. It's the entropy of x and the entropy of x after I tell you y, right? So the entropy of x is the average of minus the log of the probability distribution, so it's log L. So the log L is right. But what's the entropy of a Gaussian? Well, it's sort of log sigma, but not quite. If you do the calculation, there's a factor. And that factor, that loss of information, is related to this is precisely this confusion. Okay? It's another way of counting the same thing. So in fact, if I think that what I need is something like log L, if I've got my sigma down to the right level, and I need and I think that I'd like to get log L over sigma bits, I'm actually missing some. And, you know, in many calculations, you wave your hands and you say, you know, 2 pi, the square root of 2 pi e is approximately 1. But, you know, pull out your calculator, you'll see that the square root of 2 pi e is not approximately 1, right? Well, it depends how good an approximation you want. But, you know, this is 6 and this is almost 3. So this is, the square root is almost 4, so you're kind of missing 2 bits. And there's only 60 rows, so, right, the log, you know, 64 is only 6 bits, right? So if you're missing 2, that's a lot. So we almost got it. Where is the missing information? So the reason I like this problem, as I say, we're not quite done with it. I'll tell you what I think the answer is. The reason I like this problem is that in physics, small quantitative discrepancies matter, right? If you measure numbers and you have a theoretical framework and the numbers don't quite fit together, you actually take that difference seriously. When you come to biology, you might say, no, it's not possible, right? I couldn't possibly do experiments accurately enough that I would take the numbers that seriously, and I don't trust my theoretical framework anyway. So in some sense, what we're doing here is we're taking this little number seriously, which in a certain, if you think about it in a certain way, is really sort of a bigger number than it should be. So what's the answer? Well, the information is not just the individual positions. One way to think about it is that the information that's being transmitted ultimately from the mother to the cells is the whole sequence of positions, right? And the statement that everybody's in the right place is the statement that they come in the right order. So what I'm really worried about is, can I transmit information about this sequence? Not just about individual positions. How much, in order to compute the information that's in the sequence, well, let me be sort of hand-wavy, it's still true that the thing I'm trying to transmit is just are numbers between 0 and L, but the noise is now not just the Gaussian fluctuation of one variable, it's the fluctuations of this whole set. And of course, if those fluctuations are correlated with each other, the entropy of that noise will be smaller. And if the correlations extend over a reasonable range, then that reduction in entropy will actually be quite large and might be enough to bridge this gap. A somewhat more pedestrian way of saying it is, I drew this picture on the assumption these are independent random numbers. If they're positively correlated with each other, in fact, if they were perfectly positively correlated with each other, right, then the fact that each one of them jitters by 1% doesn't matter because they never get out of order, because their fluctuations are perfectly correlated. Okay, they're not going to be perfectly correlated, but what you can see is they're correlated enough, then you'll never make these mistakes. So now what you do is you go back to your experimentalist friends and you say, do you trust that you can measure the positions of the peaks accurately enough? You told me it was 1%. Now what I'm going to do is I'm going to notice that in embryo number 1, this guy is a little bit to the left, this guy's a little bit to the right, this guy's, and let me compute the correlations in the errors of these positions. And what you discover is that they're correlated. They're correlated over nearly a quarter of the length of the embryo. The different stripes, right, the different colors here reflect different molecules. They're correlated with each other in ways that depend on the distance between the peaks, but not the identity of the molecules. Which means that although they're being produced by different processes, they're in some sense reflecting the same underlying positional signal. And if you turn the crank and ask yourself how much is the entropy reduced, the answer is almost exactly the amount you need in order to make up the difference that we saw here. So we think that this is a case where the precision, you know, there's sort of the first order precision of seeing the absolute errors being 1%. And then there's another layer beyond that those errors are correlated in a way that makes it possible to transmit a pattern almost perfectly. And we think that we can see reflections of the same correlations already here. Okay, So this is what I haven't told you about, which is in some ways the theoretically most challenging part, but it's also the part that maybe for related reasons we're the farthest from having a complete answer, is can you think about the design, if you will, of the network of genetic regulatory interactions that happen inside this box, which is at least black inside the box, it's got an outline. Can you think about the parameters of that network and how they can be tuned by evolution to maximize the information flow through the system? And is that a theory of what's happening inside this box? So we've been making progress toward that, but I can't quite tell you the answer yet. Okay, so this was one example of precision in a biological system. And you see that it's an example where we see the precision sort of at many different places. They're all connected to each other. There are lots of interesting theoretical questions. I've kept the theoretical discussion relatively elementary. There are other examples, as I mentioned, but this is one where we've had this wonderful also interplay between theory and experiment now going on for a decade. So let me pause here, and I'm going to switch and talk about emergence. And I have some questions. Yes?
How physically distinguishable are the different segments except for the first and last one? If I switch two of those segments, will the caterpillar die? Or?
Depends at what's, I mean, the answer to that always depends on what stage you do it. So it's a classic, this is a hard experiment to do in flies, but in other organisms, there are classic embryology experiments where you take cells from one region of the embryo and exchange them with others. And so the usual view is that if you do that early enough, everything's fine. But if you wait too long, it screws up. But here, it's special, right? Because, as you say, the segments are repeated, you might think that they would be more interchangeable. And I think the answer is that the differences in the segments, so the differences in the segments are subtle, but real and functional. And so you would create problems. Yes?
You said we can count the way the cells and the way the mom broke the cell.
Yes.
The question I would make is, there is any relation with the way the mom broke the symmetry with the pattern I see, or any observable effect?
So, for example, oh yes, I mean, so this concentration profile, which is the signal that drives everything, well, it and its complementary one coming from the other side, is a direct consequence of this molecule being here. And furthermore, the mother, remember that in your DNA, right, you get one piece from your father and one piece from your mother. So that means that most of the molecules that you have two copies of the gene. So when it comes time to make this, okay, this depends on exactly which process you're talking about, but let me give you a kind of rough statement. You've got both copies, you use both of them. So that means that these molecules that are put here are coming from both, they're made from two copies of the gene for this molecule. So you can imagine that there's a fairly easy strategy for getting rid of one of them, right? So you make mutants on one side or the other, or you make mutant males that don't carry this gene, right? Now the mother only has one copy. So she makes half as much of this messenger RNA, which makes half as much of this, and these boundaries move. That's actually how this molecule was found. Yeah, so I should say that, right? The reason we know that these are the molecules that are involved is because if you have a mutation in any one of these, you change the pattern in the organism at the end. That's how they were found. Okay, let's talk about emergence for a little bit. So, I think I have time to do 2 examples. So I'm going to do the smallest scale and the largest scale. So I'll skip neurons. Many of you are probably interested in them, but we can talk later. So I told you that in biology, the molecules themselves are big enough that their structure and function are themselves emergent properties. So a protein is a polymer of amino acids, and it has a sequence that I can think of as being amino acid 1, amino acid 2, amino acid N. N is typically measured in the hundreds. You can find things which are only 50, but you can find things that are thousands. There are 20 amino acids. So if you want, you can think of the sequence as a set of variables Si alpha, and Si alpha is equal to 1 if the amino acid at I is of type alpha, and 0 otherwise. Right? So now, it's also true about proteins that unlike most polymers, they fold up into a reasonably well-defined structure. So you start with a chain, right? And then this folds, and when it folds, it becomes that, okay? Now, it's not frozen, right? So everything's still jiggling, but it's jiggling by fractions of an angstrom, right? So there's a well-defined structure. And in fact, as most of you probably know, the structures are sufficiently well-defined that you can make crystals out of them and solve their structure by X-ray diffraction. Okay, so that's how we know the structures of many of these molecules. So this problem, how do I get, another important point, is that the structure is determined by the sequence. So these 20 different amino acids, I don't want to do chemistry here. In fact, I actually can't do this. I can't draw these things from memory. It's some intrinsic cultural thing about physics that prohibits me from remembering chemical structure, molecular structures. So they're different from each other. Some of them are charged. Some of the charged ones are positive. Some of them are negative. Some of them, instead of being charged, have big aromatic rings hanging off of them that don't like being in the water. And so when the structure forms, they get buried in the interior, okay? So they have different chemical types, and it's very easy to see that some of these have the property that you put those 2 amino acids next to each other, they'll be energetically very happy. So a positive one and a negative one, okay? You don't need, thankfully, we don't need chemistry to tell us that that one will work, right? And then if you take 2 that have big rings hanging off of them, the rings can stack on each other and get away from the water, and they'll be happy, okay? So that's called the hydrophobic effect. And the process by which the sequence, the polymer folds into a well-defined structure, so this is called the protein folding problem. And there's a very practical interest in the protein folding problem, which is that if I give you the sequence, I would like to know what this molecule is. And what it does is largely determined by its structure. So if I could look at the sequence and draw the structure, I could actually figure out what this molecule did. Okay, and that would be very useful. Conceptually, right, so that's, if you want, the biochemist's version of the protein folding problem. For a physicist, the protein folding problem is different. Why is there a well-defined structure at all? If you take a random heteropolymer, what happens when you cool it down? The answer is it becomes a glass, right? Because I have a sequence in which I have randomly positioned elements, some of which like to be together, some of which like to be apart, and they're constrained by the fact that they're attached along the sequence. There are many different configurations that are all approximately degenerate. And there are lots of barriers between them, right? Once I start folding something up, it's very hard to get undone and recombine. So the result is that random heteropolymers are examples of glassy physics. And that problem you can do. It's a nice calculation. Okay. Proteins are not glassy in that sense. They have well-defined structures. So then you say, okay, that's good. So I guess, so there's, in fact, there's the idea that the way in which sequences have been selected by evolution is to minimize the frustration, right? So if you think about it, by putting the different amino acids in the right order, I can make sure that at least when the thing folds into its best structure, there are no unsatisfied interactions. So in a glass, or in a spin glass, which is easier to think about, right, you know that any locally stable state that you find has the property that some fraction of the interactions are not satisfied. That's what it means to be frustrated. So you could look at the structure of the protein and imagine that what has happened over the course of evolution is that in the real structure, all of the interactions are positive and are favorable. And that constrains the sequence enormously. It also means that if frustration isn't important, you could make a hypothetical model in which every amino acid knows who its partner is supposed to be and just get pulled toward it by some spring. And that actually gives you a theory of the dynamics of folding, which is surprisingly precise. Okay? So that suggests that the sequences that you see in nature have been selected to solve a physics problem, which is in fact essentially to avoid having this be part of the physics of glasses. So there was a period, this is another interesting example, right? It's a little bit like jumping, making the connection to non-equilibrium pattern formation because you saw a periodic pattern. There was a period in which people said, oh, complex is always equal to random. And since I have these complicated heteropolymers in biology, that must be like the random heteropolymer, and proteins should be like glasses. And indeed, there are aspects of the dynamics that if you do the right thing, you can convince yourself it looks a little bit glassy. But there's a qualitative error in that view, and that is that a real random heteropolymer has no well-defined ground state structure that can be programmed in by changing the sequence, right? Which is how proteins work. So that tells you that the sequences are constrained. So how strongly are they constrained? Oh, by the way, this process, the protein folding problem, is unambiguously an equilibrium statistical mechanics problem. There are a few cases in which the structures that you see are not in equilibrium, and we know about those. But you can make a lot of progress by thinking about this as an equilibrium statistical mechanics problem. It's an interesting equilibrium statistical mechanics problem. But there's another part to the problem, right? Which is that there are certain functions I'd like to achieve inside the cell. I have a protein that does it. And then over the course of evolutionary time, the sequence of that protein changes. And so I can look across the whole tree of life, and I can find molecules which are effectively the same molecule with slight variations that carry out the same function in different organisms, and they have slightly different sequences. Sometimes they have drastically different sequences. There's a famous example of two proteins that have almost no amino acids in common except for 3, which are actually directly involved in the chemistry. So you can't change those. And they fold into the same structure so that the backbones of the polymers can be superposed to angstrom resolution. So you wouldn't actually see that these were the same molecules, but they fold into the same structure. So in that evolutionary sampling of sequences, what are the rules? What is the ensemble of sequences that I can generate that would be consistent with making a protein that belongs to a given family? So what you could say is the usual folding problem is to tell me about the probability distribution of the structures of the molecule given that you know the sequence. And the answer is right? That, if you ask your chemist friends, they know how to construct this energy function, and it really is the Boltzmann distribution, and so, I mean, I may or may not know how to make any progress on calculating anything, but at least I know what I'm supposed to be doing. But evolutionary dynamics is different, right? It says that over the course of evolutionary history, there are mutations and selection and mutations and selection. And if I look across all of the organisms that are alive today, if they all have proteins that carry out the same function, they probably have very similar structures. And in some cases, you can directly measure the structures and show that they're the same. And so that evolutionary process is sampling some probability distribution, which is the other way, right? It's the distribution of sequences that are consistent with the particular structure. And we know that this is not a uniform distribution, right? I told you that if you randomly sample sequences, they don't fold into proteins at all. And by the way, that's not just a theoretical result. You actually try it. If you make random sequences and you get organisms to make them and you try to purify it, you ended up with a glop at the bottom of the test tube. They have not folded into well-defined structures. In fact, they're all sticking to each other. In fact, we also know that not every detail of the sequence matters. Because if I look across the tree of life, I can see that there are two molecules that have essentially the same structure, but they have different sequences. So the first idea is, well, the reason that this thing has the right structure is because if I count from 1 to 300 along the length, and I go to site number 72, it's very important to put a histidine at site number 72, which is one of the 20 possibilities. And at site number 83, it's important to put an alanine and so on. And so what that says is that you should keep track of the average or the probability of finding a particular amino acid at a particular site. And that if you obeyed those probabilities, right, at some sites it doesn't seem to matter, you put anything there. So if I obeyed those probabilities at the sites where I always put a histidine, at the sites where it doesn't matter, choose at random, but choose from the real distributions you see in nature, then you could make a protein. Right? So what is the simplest probability distribution you can write down that will reproduce these expectation values? The answer would be that, right, these are, if you want, Potts spins, right, Potts variables, right, because there's 20 possibilities at every site, but only one of them is non-zero. And so we break the symmetry among them, because at this site we like type number 3, and at this site we like type number 4, and at that site we don't care, so we restore the symmetry. So we put fields at every site, right? So that, of course, and of course these are just those rewritten. So let me go in the lab and pull sequences, actually let me go to the computer, pull sequences out of this distribution, and then go in the lab and make those molecules. Doesn't work. None of them, you make thousands of molecules, none of them are functional proteins. So then you say, okay, I know why. When everything folds up, here's amino acid number 14, here's amino acid number 73. When they fold up, they're right next to each other. So if I change, it's okay to change amino acid number 14, but for example, if I change it from being positive to negative, I better make the compensating change in this other amino acid, which is sitting right next to it, or else the whole thing's going to blow up. Slightly more subtle, if I change to a bigger amino acid, maybe I should make this one smaller so that it gets out of the way and I don't have problems packing everything together. So you think, aha, it's okay to make substitutions, but the substitutions have to be correlated. So maybe I should also keep track of the correlations. So now you say, well, what is the probability distribution that reproduces this and this? And the answer is, well, I mean, there's an infinite number of probability distributions that can reproduce these, but there's a natural choice, which is to choose the one that doesn't have any extra structure, right? So the sequences will be as random as they can possibly be, except that they have to get the first moments and the second moments right. And so that problem of finding a probability distribution that has the least possible structure but is consistent with certain measured expectation values, the way you want to do that is to maximize the entropy of the distribution while holding these expectation values fixed. This is the solution if you only hold these expectation values fixed, although this is so obvious that you don't usually need that argument. This is a little more subtle, and some of you probably know the answer. The answer is that you have an interacting Potts model, and there are couplings among the different Potts variables at different sites, and the structure of those couplings has to be chosen so that you reproduce the observed correlations. Now, there's a whole set of technical questions about whether you actually have enough data to measure these things accurately. This isn't so bad, right? This is, if there are n sites and 20 possible amino acids, then this is 20 times n, actually 19, because it has to be something. But this is bigger, right? This is 400 times n squared in principle. So you should worry. But I don't want to spend the last 15 minutes talking about those worries. So, okay. It's interesting, but the fantastic thing is that somebody tried this. They constructed this. Actually, they didn't construct this. They constructed an algorithm that draws samples out of this by a strategy which was not obviously equivalent to maximum entropy, but it was. This is an experimentalist, right? So what he was going to do was he was going to make these sequences. So the fact that he wasn't completely clear about the theory, you can forgive him, because he was going to go to the trouble of actually doing the experiment. Turns out what he was doing was drawing samples out of the maximum entropy distribution. And what's amazing is that he did it for proteins that are relatively small. They're about 50 amino acids in length. And like 2/3 of the proteins that he made worked. Now remember, 50 isn't quite the thermodynamic limit, but e to the minus 50 is a very small number. So if there was some what you expect, right, you have to get a whole bunch of things right. If there's n of them, as n becomes large, the fact that a macroscopic fraction of the things are right means you're on the right track, right? So, and that's only by keeping track of the pairwise correlations. So this is a problem which is still, people are still working on very actively. They're trying, for all the reasons that I didn't want to talk about limits of data and so on, really doing this right is still hard. And you can even argue about whether it was done right the first time. But there's two different directions in which I think this is interesting. One is, again, a practical direction. The intuition is that the origin of these interactions is because the amino acids are touching each other and interacting physically. But remember, this thing that looks like a Hamiltonian sitting up in the exponential is not the real, it's not this Hamiltonian, right? This is the result of this evolutionary process. But nonetheless, you have the intuition that they're somehow connected. And you might guess that what really matters is whether two amino acids that are next to each other are interacting. If you look at this pattern of correlations, and you ask, are the correlations primarily between amino acids that are next to each other? The answer is no. But of course you know this, because if you do statistical mechanics and you have local interactions, you can produce correlations that have any range you want. If you're at the critical point, the range, right, the range is the size of the sample. And so the correlations can extend over distances which are much larger than the interactions. So another remarkable result is if you go through this exercise and you look at the structure of these interactions, you discover that these are much more nearly short-ranged than these. And in particular, the largest elements of this matrix are the amino acids that are closest together. And what that means, and there's a gap, so you can find them. And what that means is that if you go through this exercise, just by looking at the statistical structure of the sequences, I can tell you which amino acids are next to each other. But I also know that they're attached along the chain. And so now I can figure out what the structure is. So that means, remember I told you the biochemist's version of the protein folding problem was I give you the sequence, tell me the structure. So here you've been able to do something which is not quite that. It's give me 500 proteins that are all related to each other. Show me the sequences. By looking at the statistical structure of the sequences, by looking at the ensemble of sequences and building a statistical mechanics for that ensemble, implicitly I've told you something about the structure, and I can pull that out. And that's a pretty amazing idea. But there's another thing which I think is really important here, which is getting less attention, but I'm hoping it'll come back, is that, you know, when I joked about how there was a community of physicists who wanted proteins to be glasses, it's only half a joke, right? If you want, there's two extreme views. Any realistic description of a biological system would have many parameters in it, right? In particular, the sequences of all the proteins, right? Your genome, if you want, is the description I need in order to explain what you're going to look like and how you're going to behave. So that's a lot of data. So one extreme view, which you might cartoon as a certain kind of biologist view, is, yep, every single one of those details is really important. I personally make my living measuring this detail, and so I know for sure that this detail must be important because otherwise I'm wasting my time. And my friend, he works on this detail, so that must be important too. And if we connect all of our friends, eventually everything's important, right? Okay. Then, you know, somewhere over on a very different direction, there's a certain species of physicist who would say, all these parameters, pick them at random. It's so complicated, it can't possibly matter. Right? So let's do something generic. Let's not worry about all those details. Okay, what's fantastic about this problem is you absolutely know by both theory and experiment that both of those extreme views are wrong. Okay? It is not true that every single detail matters because you can go and you don't have to wait for evolution to have done the experiment. You can go into the protein, manipulate the sequence, and get the same structure back out with a different set of amino acids. So obviously it was not important that it's exactly alanine at that particular site because I can replace it with a glycine and it still works. On the other hand, I also know that if I draw things completely at random, I don't get anything that's even qualitatively like real proteins. So the real system sits somewhere in between. The ensemble of, if you want, legal biological systems is not trivial. It is neither exactly what you see and nothing else, nor anything goes. There's something in between. And you could argue that many of the problems that we're interested in, the essential question is to define that ensemble. So for instance, I want to build a network of neurons. I want that network of neurons to carry out some function. I might want this to be a model for what's going on in some real brain, or I might want it as a computing device to do some problem in artificial intelligence. The parameters of the system are the strengths of the connections among all the neurons, the synaptic weights, if you use the language of artificial neural networks. There are a large number of those. Do I need to know every single value? That can't be right. Can I pick them at random? Well, it's very easy to show that that doesn't work. There's some non-trivial distribution over this space of parameters that's consistent with actually performing the function that you care about. And that object is a very fundamental object in our understanding of how this whole process works. And you can ask it again, not in the context of neural networks, but in the context of genetic networks. Is the fact that this particular protein binds to exactly 7 sites along the DNA that are close to the place where this gene gets read out, is 7 important? Is how strongly it binds to every one important? Is it only certain combinations of them? Can I imagine an ensemble of possible flies, right, that accomplish all this with slightly different values of all these parameters? Maybe that ensemble is the set of things that you see over evolutionary history, which would be really nice, but maybe it's just things that are possible and I could go in the lab and make them. As a physicist, I'm interested not in the particular, but in the general, and I'm given particular examples. So what I want to imagine is the distribution out of which those things are being drawn. And I think This problem is interesting because it's a place where we're getting very close to being able to write that distribution down. Okay? And I suspect we might get to it here before we get to it in some of the other examples. It's a little bit simpler in some ways. Okay? But the same question arises. I think the neural network example is one that's very much in people's minds because we know that these artificial neural networks are capable of solving problems that we thought were incredibly hard, like face recognition. They reach near human level performance, and we don't understand why they work. And we know that the learning process for training those networks is such that every time you do it, you're going to get a different set of synaptic weights. So there is some distribution. There is not a single answer. And so having a theory of how that works is having a theory of that distribution. Okay. Having a theory of protein structure and function is having a theory of this distribution. So one step is, can I just characterize the distribution? Can I infer things about the distribution from the data? But of course, there's another problem, which is to explain why it would have this form from some reasonable model of evolutionary dynamics and so on. Knowing that, of course, there's real physics in the sense of mechanics that stands behind it. Right? And what's the relationship between this function and that function? Okay, let me give one more example of emergence. I told you I'd do the smallest scale. Let's do the biggest scale. Let's think about a flock of birds. So in a flock of birds, depending on exactly which flock we're talking about, or which kind of birds. So many of you have seen birds flying in formation. So, you know, when the geese all line up and they do things, right? That's not what I'm talking about. That's flying in formation, not flocking. So think about things like starlings, where you see these they're ordered in the sense that almost all the birds are going in the same direction, but they're also fluid because the direction changes, the flock can expand, it can contract. When a predator arrives, the whole thing can turn and go off in a different direction. Okay, it's that kind of thing that we're talking about. And again, this subject has been revolutionized by the fact that it's possible to do multi-camera imaging of large flocks, and by doing very clever analysis, you can reconstruct the three-dimensional positions of thousands of birds in a single flock. Okay, and the heroes of this subject are my colleagues in Rome, Andrea Cavagna and Irena Giardina, who actually started their careers doing statistical physics. And I guess you would argue they're still doing statistical physics, just now on birds. This really sort of changed the whole discussion. There had been theories of flocking and schooling and swarming, both very simple computational models. There were attempts to take those computational models, and if you think that the birds are like particles, then if you have a whole lot of them and you zoom out, you'd actually like to study the hydrodynamics of the particles, right? I don't want to do molecular dynamics, I want to do fluid mechanics. So can I find the field theory that describes the hydrodynamics of these flocks? And okay, this is a very serious enterprise, quite sophisticated. But there was no data. Now there's data. So let me ask a simple question. And to make it as connected as possible to the discussion we had here, which also saves time. I show you a collection of birds, and each bird has a velocity v. It's a vector. You know that they're aligned, so they're all pointing in more or less the same direction, although not exactly. And can I describe for you the probability distribution of those velocities? So I show you a snapshot of what all the velocities are. Of course, it's really a dynamical problem, so we could ask other questions, but to keep it as close to this as possible, let's imagine I show you, here, of course, I could have asked a dynamical question too, right? I could have asked about I could ask specifically about the ancestors of particular organisms and how those sequences are related to each other. But I didn't, I just said take a slice. So let's do the same thing here. I take a snapshot, I see all these velocities. Can I write down the probability distribution of all the velocities? Okay, well, what do you think is important about the flock of birds? You think that birds are looking to their neighbors and trying to make their velocities similar to those of their neighbors. They're not, one suspects, although you don't actually know this when you start, they're not looking all the way across the flock at the guy on the other side to do what he's doing, right? They're looking at their neighbors. So if that's true, then the crucial index of similarity, right, the crucial index that characterizes the mechanisms that ultimately give rise to ordering the flocks is some local correlation, or some local ordering. So I could look at the velocity of one particular bird, and I could look in some neighborhood So let me define a neighborhood of each bird, and let me agree that the size of the neighborhood is some number of birds. So let's draw a circle here, and the neighborhood is all the birds in that circle surrounding the guy I care about. And let me compare my velocity to the average of the birds in my neighborhood. And let's average this over all of the birds and compute the square, right? So I measure my mean square deviation. So this is an index of how well I'm doing at behaving like my neighbors. And it's a local quantity, right? It focuses on the belief that the interactions are fundamentally local. But you realize, of course, that if everybody flies faster, Q stays the same, right? If everybody just gets boosted. So I should keep track of a few other things, and I'm going to do this in the way that we did it when we first started, which is slightly awkward, so I apologize for this. You could think about the average speed at which all the birds are flying. Of course, that's just a matter of units, right? So I could call that 1 if I want to. But you also know that birds don't just fly at some average speed. They also, their speed fluctuates. They try to be like their neighbors, but their speed fluctuates. So let me insist that you also keep track of the mean square speed. So now in the same spirit that when I was thinking about proteins, if I told you about one-body objects and two-body objects, I could construct the maximum entropy distribution that's consistent with this. I can do the same thing here. I can ask you, what is the minimally structured theory of the distribution of velocities, the joint distribution of velocities in the entire flock of birds that is consistent with these objects, right, with these measurements on the behavior of the flock. And remember that because these measurements are averaged over the entire flock, right, they're like measuring expectation values. In fact, to make that clearer, I should have put a 1 over n here. So what is the maximum entropy distribution that's consistent with these? Well, you see the structure. It's this, right? So same argument, right? In order to generate the maximum entropy distribution, right, you need to maximize the entropy, but you need Lagrange multipliers that hold each of the expectation values constant. Those Lagrange multipliers end up in the exponential, and that is the maximum entropy distribution, right? Actually, how many of you understand what I just said? Okay, how about this one? Good. If I ask you for the maximum entropy distribution, a distribution over variables that maximizes the entropy while holding the average energy of the system fixed, what's the answer? It's Boltzmann distribution, right? You'll notice that it's e to the minus a Lagrange multiplier times the thing whose expectation value you held fixed. Now, the fact that in the Boltzmann distribution this is the energy, the same energy that it is in mechanics, is of course incredibly fundamental. However, if you make the argument, give me the maximum entropy distribution consistent with the average value of the energy, the fact that it was the energy that you constrained is not essential. So you could have constrained something else, and then that was the thing that would appear up here. And if you constrain several things, you put multiple things up here. And so that's why this is the answer. Okay. And you'll notice that this looks exactly like a Boltzmann distribution, where this is the energy. And you can determine these parameters just by fixing these values. And let me emphasize that these values are expectation values of local quantities in the flock. Okay? So now, is this theory right? Well, with proteins, the way I found out whether the theory was right is draw samples out of the distribution and make the protein. It's pretty cool. I don't know how to do that with a flock of birds. So what I'm going to do is I'm going to draw samples out of the distribution and compute their statistical properties and ask whether those correspond to the statistical properties of the flocks that I see. So what's the most obvious thing to do? Compute the correlation function of the velocities. Remember that the only thing you used was the correlation with your immediate neighbors. So, but the flock is thousands of birds. So there are distances which are 10, 20, 30 times larger than the distance to your neighbors. So I have a whole correlation function I can try to predict. A remarkable feature of real starling flocks is that those correlations are long-ranged and essentially scale-free. To the extent that you think they have any structure on a particular scale, if you find yourself a flock which is twice as large in linear dimensions, all of those correlation lengths change by a factor of 2. So there's a non-trivial structure that you have to predict, and you get it right within error bars. And the error bars are not that big. I could sketch them for you, I could show you the real data, but it's remarkable that this theory, which is relatively simple and fixes only local quantities, only takes local quantities from the data as input, gives you a parameter-free prediction for what the entire correlation function should be, and it's right all the way out to the longest distances corresponding to the size of the flock. Furthermore, if you do the same analysis in different size flocks, you see the scale invariance. Then you ask yourself, why on earth is this happening? Take this theory and butcher it to make a continuum limit out of it. And what you have, of course, is some sort of Heisenberg model, right? Because you have these vectors that can point in any direction. They have a direction. Well, sorry, it's not. Let me back up a second. These are vector velocities, so they have an amplitude and a direction. Suppose I only thought about the direction. Then in that case, I have a Heisenberg model. The couplings to my neighbors are fairly strong. Because they're fairly strong, the whole flock polarizes. I break the symmetry spontaneously. If I break a spontaneous symmetry, if I spontaneously break a continuous symmetry in a theory that has local interactions, I have Goldstone modes. So I should see long-range correlations of the directions of flight because of Goldstone's theorem. And that's true. That's what you see. The more subtle problem is the fluctuations in the amplitude, because there's no symmetry. Okay. In fact, what you find is that the family of theories that we've written down actually has a critical point at which the fluctuations in the amplitude would also have divergent correlation length. And the values of these parameters, which are driven by these measurements, are very close to that critical point. And that's why you see these long-range correlations. Okay. This theory, as I said, there's reasons why this is not what we should have written down, because the critical point in this case is actually at an edge of the parameter space, but that is actually where you're driven. You could do a better job, but I'm trying to be faithful to what we actually did the first time around. So what I hope to have illustrated for you is that in systems ranging from single protein molecules to the complicated genetic networks that drive embryonic development, all the way up to thinking about large groups of animals, we're at a point where we can make comparisons between theory and experiment which are much more precise than we could have imagined a decade ago. And the phenomena that we see that you can study quantitatively in these experiments are the kinds of surprising things that in many other areas of physics have deep theoretical explanations. And so, is it the case that somehow groups of organisms organize themselves to operate near a critical point in the space of possible flocks, and that this is there to allow, for instance, for long-range communication, right? The long-range correlations that emerge as you approach the critical point have as a corollary the ability of the flock to transmit information throughout the flock over long distances, and that's important for avoiding predators. So maybe the things which are striking to us as physicists are also actually connected to the biological function. In the example of proteins, you see that questions which are natural from a physics point of view really are very basic problems about the overall diversity of molecules that you can make. If you ask this question not about proteins in general, but only about the proteins that you use for making antibodies in your immune system, then the diversity of antibodies, right, the distribution that you're writing down directly characterizes the distribution of molecules that you make as you try to fight off infections. And that distribution is characteristic of the functionality of the system as a whole. So what I think is exciting is not only that you're starting to see glimpses of problems that have some structure to them from a physicist's point of view, but that those problems are tied to what makes the biological system special, right? It's not the generic example of pattern formation. It's not the generic example of self-propelled particles. It's, real patterns forming in an embryo or real flocks of birds. And those have properties which are related to the generic case, but somehow special in ways that we can start to get our hands on, and that point to us toward different physics than you might have guessed if you hadn't looked at real life. So, thanks. So, let me make an observation. There's a 2-hour lunch period. I'm only here today. So for some fraction of this 2-hour lunch period, I'm going to sit here, and if you guys want to talk with me, you want to talk about physics, you want to talk about things in the lecture, you want to talk about how things work in Princeton, please come. I don't know if there are a few questions, we can take them now, but let's say I will definitely come back here at 2, which I guess leaves an hour before you start lecturing in the afternoon. So if there's a question, I'm happy, but you can also come back.
So we'll take questions now. And
Okay.
We'll go back. So I should tell you before I forget, on Saturday and Sunday, most restaurants are closed. So today the cafeteria just near our building is open. So that doesn't have much variety. But there's another place where there's a lot of variety, which I think you got the email for the instructions. But if you just follow everybody, you go there too. So 5-minute walk. And tomorrow, just that place. The cafeteria is closed tomorrow. So, any questions for Bill?
Maybe you'll be fortified after lunch. Yes? Ah, very good. First initial, last name. Yeah. And yeah, feel free. It might take a day, but I do try to respond. Yeah.
Just a naive question. I was reading that the DeepMind was folding proteins.
Yes, I don't know exactly what they're doing. So So as I say, in thinking about the protein folding problem, as opposed to this, which is sort of the inverse folding problem, there's one approach is you say, well, look, you know, it's equilibrium statistical mechanics. I know how to write this down, or at least my chemist friends tell me that they know how to write this down. So let's just go and compute, right? Of course, that's very hard, because these are very big systems. So then what people do is they say, here's a short segment of 5 amino acids. If I look through all the proteins whose structures I know, hey, exactly those 5 amino acids appear in these other 3 proteins. And every time, it makes one of these helical structures. So maybe this part's a helix. And maybe if I do enough of those, I can piece together an estimate of the whole structure. That's the thing which sort of kind of works, not so well. But it's also the direction where you can imagine that modern machine learning techniques could do much better, right? Because instead of saying, oh, I found this particular sequence and I found an exact match, and then you say, oh, maybe I don't need an exact match, so I make up some function that tells me how close the match has to be. You say, well, try to learn all that in some systematic way. So people are trying to do that. And yeah, but that's about solving, in some sense, the practical protein folding problem. I give you the sequence, draw me the structure. The deeper question, I think, is what is it about these sequences that allows for a structure at all? Yeah. Maybe eventually it'll trigger them to ask one.
I would imagine that in these problems where you have a macroscopic scale and then a macroscopic scale about the
Yes.
density of birds and so on, that often the right variables to describe what happens at large distances have little to do with the microscopic variables, right? As in field theory,
Yes.
the variables that describe the infrared and
Yes.
particles are not the quarks that
Yes.
describe the microscopic. So why wouldn't we expect something similar here? Why would we expect that the velocities, the microscopic, would be the relevant degrees of freedom for the macroscopic? And in particular, if I look at this probability, it looks like some terms here are relevant, so they would flow in the infrared, and I would lose control over what I'm doing. Like a second.
Yes. So, great question. So, long before there were measurements, John Toner and Yuhai Tu tried to do exactly what you're describing. They said, let me take a model of particles that always move at a constant velocity, and they do something relative to their neighbors, and let me go through the full machinery of doing the coarse graining and doing the RG for this dynamic theory and get the theory in the infrared. So, a fascinating fact is, of course, you started with some microscopic model which might or might not be in the same universality class as real flux. So that's not a criticism, it's just you have to remember that. Many of the things that you get as generic predictions of that theory turn out not to be true about real flux. Maybe about some other groups of organisms, but not about real flux. The most dramatic one is something that I haven't talked about here, but is very much something that my friends in Rome think about, which is that all of the theories that people wrote down had a dynamics which was essentially diffusive. So if you ask, you know, if you say that everybody orients to their neighbors, to the average of their neighbors, right? If you have a variable which over time tries to become like the average of its neighbors, you're describing diffusion. Okay? So that would suggest that if one bird starts trying to turn, that information would spread through the flock diffusively. The front of birds turning would have a radius that grows like the square root of the time. In fact, if you observe groups of birds turning, what you find is that the information propagates through the flock ballistically. So it is as if somehow there's an inertia. But you know that if you write down a theory of inertia at the microscopic scale and there's any dissipation, then in the real infrared limit, the inertia will go away, right? Because you'll lose whatever sort of almost momentum conservation or something you had that gave you this inertia will be lost by coarse graining. So somehow real flocks avoid this. I mean, it's not that if you coarse grain, that isn't what happens. It's just the question is on what scale do you cross over to get between these different things? And so what I think is fascinating about the real flocks is that if you try to think about this, I write down something microscopic, I coarse grain, I see what I should get. It's like somehow the flocks have managed to position themselves in a way where they can sort of squeeze a lot out of certain microscopic things before they take advantage of certain irrelevant operators before they've been completely scaled away. And that, I mean, there's something intriguing about that, right? How do you position yourself at that point? Because it's somehow not a very generic point. I don't know if that's so that's one answer. There's another answer. If I didn't know fluid mechanics, but I gave you a movie of what the molecules were doing, you would start there and try and analyze it. One of the things you might try to do is to coarse grain, although how you knew to coarse grain but didn't know fluid mechanics is an interesting historical question. But so a different approach would be to say, let me take the real data and try and coarse grain it. In the flocks, we're trying to do that, and we haven't gotten there. In the case of neurons, we actually have been able to do that with really quite startling results. So you see that the distributions seem to be flowing to some non-trivial fixed point and things like this. So yes, I think one of the frontiers of dealing with this very large data is to try to take our understanding. Actually, let me say this in a way that's maybe more useful to all the students who don't know some of these things. One of the really important things that we've learned in physics is that our description of the macroscopic world is simpler than it could be, not because there's something special that happens, but because in the flow from microscopic to macroscopic, there are lots of irrelevant operators that go away, okay? And so if you start with a complicated microscopic description, many of those details will disappear, and correspondingly, you can have a successful description of the macroscopic physics that is not at all faithful to the microscopic details, right? That emergence of simplification that we understand through the renormalization group, at least in some set of problems, right, is an incredible inspiration for thinking about much more complex systems, because it gives you the hope that you can reproduce that success in this more complex context. The problem is that we don't really know how to do that. It's at the moment just an inspiration, and so people are feeling their way. But as we enter an era with much larger data sets, the question of whether that insight will be directly useful in analyzing those data, I think, is a very exciting possibility. That wasn't a terrible place to stop. I'm hungry. Let's